Good morning, Mary and Nathan. We'll give things a few minutes here as people start to connect in.
Morning.
Push this again. Thank you, everybody, for joining us today. This is the second day of the RIT in Africa Research Colloquium. We are really excited to present this year from our RIT Global Office and appreciate everybody who is engaging in hearing the research of these wonderful RIT faculty and PhD students. So this morning with us, we have Dr. Mary Obo, who is a postdoc researcher at RIT in the College of Health Sciences and Technology. And she will be presenting the interaction of tumor necrosis factor alpha and malaria pathological outcomes this morning. So with that, Mary, I turn it over to you to start us off.
Thank you, Lindsay. And good morning, everyone. As Lindsay has rightly said, my name is Mary Obo. I would be presenting on interaction between a particular human gene called tumor necrosis factor alpha and the malaria clinical, in other words, pathological outcome. I am in the College of Health Sciences with Bolaji Thomas as my supervisor. Malaria, I'll start by introducing us to what malaria is and some epidemiological statistics about the disease. Malaria is one of the major parasitic diseases that we have, and it results into substantial morbidity and mortality, especially in the tropics and subtropical regions of the world. In 2020 alone, the World Health Organization estimated that there are about 241 million cases of malaria. Now, of these 241 million cases, 95% of them were from the African region. It goes to show the burden of this infection in Africa. Now, Nigeria accounted for 27% of the global cases. So of these 241 million, Nigeria contributes the highest in terms of malaria burden globally. Now, the disease malaria is caused by a species, a genus that is called Plasmodium. The parasite is called Plasmodium. And we have several species that causes these malaria. But one of the most critical, that is the most deadly of them, is the Plasmodium falciparum. And this is the one that is widely distributed in Africa. So Plasmodium falciparum, it results in a wide spectrum of pathological outcomes, from individuals who are infected with this parasite and yet remain asymptomatic, to those who become symptomatic but do not have any complication, and to the more very severe and cerebral conditions, which in most cases becomes fatal for anybody that gets infected with this disease, this condition. Now, there are different factors that actually affect the outcome of malaria. From the parasite factors, we would look at the virulence of the parasite, the strains of the parasite, and as well as the parasite genetics. We also have the host factors in terms of the host now being human, because we have the human and non-human host. But in this case, I'm referring to the human host. So the host factors here specifically looks at genetics and the immune responses in individuals who become infected. And we also have the drugs and the pharmacokinetics as part of what also affects the outcome of malaria. But in this case, I would be looking more at the host factors now, in this case, genetics as well as immune response. We have different studies that have provided evidences on how some immune responses affect and lead to severe condition. Now, we have what we call the pro and anti-inflammatory cytokines. Now, these are immune responses that actually determines how malaria infection progresses and what the clinical manifestation would be. This particular gene of interest I'm looking at, which is called the tumor necrosis factor, is a pro-inflammatory cytokines. Pro in the sense that it acts as the first line of defense against infection with Plasmodium falciparum. Now, it acts in a cascade of different ways. First of all, it's secreted by what we call the macrophages. Macrophages are small white blood cells that are produced when a foreign agent, in this case, the foreign agent would be malaria parasite or Plasmodium, enters into the body. So the tumor necrosis factor acts as a pro-inflammatory factor. Now, in doing that, it also activates other pro-inflammatory cytokines. In other words, we say that it is seeking for help or assistance from other immune cells to come and help it in clearing the infection. Now, in doing that, it prevents the growth of the parasite, but it also acts as a double-edged sword, whereby as it tends to secrete more or become expressed, there's what we call expression of immune cells. In a bid to try to suppress parasite growth in the host, it becomes highly expressed. Now, in that process of being highly expressed and clearing the parasite, it's also causing a damage to the human cells. So that's why we say it's a double-edged sword. Why it tries to bring down the infection at higher expression level, it also does harm to the body. So some studies use the expression level of TNF alpha, tumor necrosis factor, to measure how severe a condition is, whereby if you have it at higher concentration, it provokes severe and even sometimes cerebral malaria, in which case you would have the parasite to have crossed the blood-brain barrier, where it is not normally supposed to do, and that becomes a fatal condition. But at lower expression level, it becomes protective of against severe condition. But how then do you determine, how then does it regulate itself so that there is a balance between overexpression and the moderate expression? And that actually has to do with some particular human genetics that affect the expression. There is a study here by Westman et al. In 2015 where they actually determined and gave evidence of how the tumor necrosis factor affects the malaria pathological outcomes. And so we can see here, if you look at figure A here, you see that from the left-hand side of the graph, you will see that when it is moderately expressed or at low level expression, there is no complication in malaria pathological outcomes. So what this result into is that you have less adhesion. So adhesion talks about the attachment of the parasitized red blood cells to blood vessels. So when you have it at low expression level, there is no much adhesion, no more attachment, as we can see here. But when it is highly expressed, as seen on the right-hand side of this graph, you have higher adhesion, it becomes overly expressed, and so parasitized red blood cells become attached to blood vessels or microvascular system, and so it begins to cause a cascade of other severe conditions. Now, this expression level of TNF-alpha is really tightly regulated. It is regulated by transcription. Transcription here talks about the process whereby a messenger RNA or a DNA becomes what we say turns, let me use a word that will be generally understand, is modified to produce a protein, right? It is a protein expression that we'll see. So this process of modification from mRNA to a protein is what we call transcription, and it is tightly regulated in this gene, this TNF-alpha gene. And because of these, we have some changes that we call polymorphism that occurs at the 5' end region of this gene. So in genes and proteins, we have what we call the 5' end and the 3' end. The 5' end is close to the promoter region, is a region where you have this transcription occurring. So this process of changing from mRNA to proteins occurs, and during this process, we have changes that we call polymorphisms that occur at this 5' end. Now, certain polymorphisms, which in other words we call single nucleotide polymorphism, have been known to occur specifically at the 5' end and have been known to impact on the production of these cytokines, this TNF-alpha gene. Now, once there is the presence of this polymorphism, it instigates more or excessive production or expression of TNF-alpha. Now, there are certain types of these variants of polymorphism that have been associated with severe condition because they initiate overexpression of this gene. So two of such variants of polymorphism that have been associated, they are what we have been identified to be the 308 adenine to adenine polymorphism. So this 308 looks at the codon, the position, the nucleotide position on the human gene. So at this position 308 from adenine to adenine, and also at this position 1031 from cytosine to cytosine, these two variants have been known to be associated with both the cerebral and severe malaria condition in Gambian and Indian children. Now, looking at these evidences before us, why then do we initiate this study? We have a number of studies which have been carried out to ascertain the role of single nucleotide polymorphisms or variants that are present in the promoter region. As I said before, the 5' end of the gene, they have been detected, to be associated with malaria pathology through genotyping. But studies have not really focused on other parts of the gene that are outside the promoter region. Do we have other SNPs or other single nucleotide polymorphism in this gene outside the promoter region that could also impact on the malaria outcomes? So this study tends to explore that in addition to the aforementioned single variants that have been seen. So the objective of this study was to analyze the association of tumor necrosis factor alpha genotype with malaria phenotypes, and also look at the possible role of natural selection that provide some form of protection and like resistance against malaria. But this study was focused more in Cross River State, Nigeria. Cross River State is located in southern Nigeria. So malaria in Nigeria is very heterogeneous in the sense that we have the country divided purely in terms of malaria endemicity into the very hyper endemic south and the meso-endemic north. So in southern Nigeria, we have malaria occurring all throughout the year, unlike in the northern part where you have malaria very seasonal, only occurs during the rainy season. So this study was conducted in that part of the country where you have malaria occurring all through the year, and particularly in Cross River State, Nigeria. We looked at children 1 to 5 years that have different clinical manifestation, whether they are asymptomatic in the sense that they have malaria, they do not produce any symptoms, and as such, there was no really justification for visiting the hospital, but they have the parasite. We also look at children with symptomatic condition. They have malaria, they have the symptoms, and we confirm this by both microscopic as well as other molecular detection tools. Then we also recruit another set of children that we call the severe group of the study and the severe cohorts. And this particular set of children have parasites that are, so according to the World Health Organization, to classify children as having severe condition, they should have more than 100,000 parasites per microliter of blood. Their hemoglobin level should be less than 7 grams per deciliter. They should have also packed cell volume that is below 20. So using this World Health Organization classification system, we're able to recruit children into these 3 arms. After we obtain parental consent, of course, because they can't give consent, we drew 1.5 to 2 ml of blood before they were administered any anti-malaria drug. These blood were spotted on filter paper because it's in field condition where electric power supply is very limited. So in order to preserve the integrity of the blood of the sample, we had to spot on filter paper. And then in the lab, we confirmed that these people actually had malaria parasite by carrying out what we call a real-time PCR molecular-based test. Now, these are schematics of the workflow. When we got to the lab, as in other basic molecular biology experiments, we isolated the DNA using the QIAGEN mini blood kit isolation kits, after which we use specific primers that target this tumor necrosis factor, to amplify this gene in a final volume of 25 microliter using this concentration of primers as well as the DNTPs with 1 unit of Taq polymerase. Now, after amplification, we had to confirm that these genes were truly amplified before we send them out for sequencing downstream. So we did that by visualizing the product on 2% agarose gel. So only those samples that amplified that we took further for downstream purification using an enzyme called ExoSAP. So after the purification, we proceed further to sequencing these products in what we call 2x coverage or bidirectionally. So when we finish the sequencing, we got result sequencing, and then we edit the results, and then we analyze in order to look for possible association of single nucleotide polymorphism with malaria clinical outcomes in the 3 groups. So here, we looked at the genotypic frequencies of single nucleotide polymorphism from tumor necrosis factor with clinical outcomes. So in this study, we saw 4 single nucleotide polymorphisms. And we can see here that the different groups of cohorts of the children actually express or show different genotypic frequencies. For example, for the severe malaria harm, we can see here that individuals with this, what we call homozygous mutants, this is homozygous. So I think I want to be a little bit basic here for our audience that might not be into molecular biology. So here, when you have this letter T, these, so they are called nucleotides. In DNA, we have 4 nucleotides: thiamines, adenine, cytosine, and guanine. So when you have the first letter here in a nucleotide depicts the wild type. Now, when you have the sign here and another letter after it, it depicts a mutant type. In other words, the change is occurring from a T to a C. So this is the wild, this is the mutants. And so because this particular gene is an autosomal gene, and what we mean by autosomal gene, it's not a sex gene, it's from the body. So it's represented in terms of allele 2, the frequency it's always 2. So here is the wild type homozygous, the same homo. Here is the mutant heterozygous. So you have a mutant, but you still have the wild type. But here is the homozygous mutant. Now, earlier I said that these homozygous mutants, especially in this and another particular codon, 308, has been associated with severe condition. In this study, we saw similar trend whereby children with severe malaria we have more of these homozygous mutants in them. Unlike children with symptomatic or asymptomatic malaria, whereby they don't have these homozygous mutants, especially in the asymptomatic condition. What these signify here is that having the homozygous wild type or the heterozygous mutant type is actually protective against severe condition, as we have seen here in both the symptomatic and the asymptomatic group, unlike here where we have majority of the participants expressing this homozygous mutant. This same trend was also observed in these different single nucleotide polymorphisms that we saw here in the study. So in order to check if the frequencies of these alleles that we have seen in table 1, of all the SNPs occurred at random. Did they just randomly occur? Or whether there was any selection pressure in any of the groups, that could actually be impacting on how this alleles occur. We carried out a chi-square test using the genotypic data of all the segregating sites. When I mean segregating sites now here, I mean the sites that had difference in terms of nucleotide position, in these 3 groups. And what we found here is that the higher number of homozygous mutants that we saw, especially in the severe malaria group at codon 1031, was actually a non-random distribution. So it's what we are saying here is that it doesn't just occur randomly and then we say, oh, it's by chance. No, it's a non-random distribution. And this means here that it is actually impacting on how the disease progresses. It actually has to do with the natural selection. And so we saw it to be significant in the severe malaria group, as denoted by the p-value that we have here, which is less than 0.01. In the symptomatic malaria group, the heterozygous and homozygous, heterozygous mutant and the homozygous wild type occurred actually in a non-random fashion, and it was statistically significant. So what this implies is that the human genetics actually is occurring not randomly, but it's occurring and impacting on the progression of the malaria clinical outcomes. We also try to check that since, this statistically significant difference expected value from the observed one indicates deviation. So this non-random observation that we saw, does it deviate from, there's a parameter we use in molecular biology, we call the Hardy-Weber equilibrium. Now, does it deviate from this equilibrium? And what we found out here is that there is one particular single nuclear polymorphism in the severe malaria group, which is the 863 mutation from cytosine to adenine. It actually deviates from the Hardy-Weber equilibrium. So the implication of this thing is that there are some factors naturally that is impacting on this gene, and that is causing the overexpression of tumor necrosis factor in children with severe malaria. We also try to understand the association between what we call haplotypes and the malaria clinical outcomes. So haplotypes just talks about group of this gene that looks similar and how do they relate to the malaria outcome. Here we saw that although, so if you look at the second column here where we have haplotype diversity, you would see here that the haplotypes in all the groups, they were highly diverse. So the diversity statistics ranges from 0 to 1. And so we can see here that for the first 2 groups, they are almost 1. And for the last group, it's more than half. So they are highly diverse, but there was no statistical difference between the haplotypes and the clinical outcomes. We also check if the tumor necrosis factor alpha is evolving under natural, we'll call natural model of evolution. Is it just occurring naturally, or we have some underlying genetics that is impacting on that? To do that, we use 2 test statistics, what we call the nucleotide diversity and the Tangeman Z test of neutrality. So in the nucleotide diversity, we looked at these two. The first one here estimates the differences between two sequences. Why these other statistics here actually estimates how many different sites do we have per sequence. So what we can tell here from our result is that the difference, there was no much differences between any two sequences in any of the group. As we can see here, the range of value for these also ranges from 0 to 1. So as you can see here, the diversity or the differences between sequences in each of these groups was very low. This was also observed for this other nucleotide diversity test. However, when we try to evaluate what is now this, what is the difference between this nucleotide diversity here and these other statistics, we use the Tangimas test of neutrality to ascertain that. We saw that there is a huge difference, between the number of sequences and the estimated segregating sites in each of the cohorts, in each of the category. But these differences, when we tested for significance, they were not really significant from each other. So what this means is that this gene is occurring under what we call a balancing selection. So yes, we have underlying human genetics impacting on the malaria pathological outcomes, but it is occurring in a somewhat, it's balancing itself out. So remember when at the background, I talked about the double-edged sword of the tumor necrosis factor, positively protecting against severe condition, but when it's overexpressed, it actually causes a deleterious effect. So there is a form of balancing selection that is controlling how this gene evolved. That is what we saw here in our study. So what can we say overall about our findings? We have reported that here that there is a possible association of, as I said, the homozygous allele at the 1031 position of the TNF alpha promoter gene with, in this case, severe malaria condition. And we also saw here that the heterozygous allele actually protect against severe condition, as we saw in table 1. The sequencing of the promoter region and the alignments with the reference strain revealed here in our study the presence of a new form of mutation that is located at codon 806, where we have a cytosine mutating into a thiamine in malaria-infected individuals. Now, the heterozygous allele, when we say heterozygous, we have a CT of this genotype. We also found it to be protective against severe malaria. So children with this heterozygous allele, CT, they were protected as against those with the homozygous mutant, a TT. In the severe group, the higher number of homozygous mutants, when compared to their wild counterparts at codon 1031, was responsible for the non-random distribution that we saw, as I explained in table 2. Now, for the same position, 1031, now in the asymptomatic group, the number of observed heterozygous and wild-type allele was higher, and the homozygous mutant was practically absent, showing the protective effect of the homozygous, wild type, and the heterozygous mutant. In this study, we observed a high frequency of homozygous rare allele at codon 863 in the severe malaria group as compared to the other groups. Our results here not only point to an association between mutation at 1031 and 863 codons with severe malaria, but it also, it demonstrates the dual nature of TNF, the protective effect, as well as the propensity to cause severe condition. This may also mean that these alleles may play a role in the progress of a disease into its severe forms, along with increasing children's susceptibility to malaria. We also observe a deviation from Hardy-Weinberg equilibrium for some of these SNPs in different clinical manifestations. And this here indicates that there is a possible role of natural selection providing protection against severe malaria, as I explained. And this particularly holds true, for mutation at codon 1031. Our results indicated possible interaction between single nucleotide polymorphism and tumor necrosis factor alpha at the promoter region, which, in children with different clinical manifestation. I would like to acknowledge my boss, Professor Bolaji Thomas, my former boss, Alfred Amamwagwa in MRC, my colleague, Amele, Dr. Amele, the European and Developing Countries Clinical Trial Partnership. I would also like to acknowledge RIT and American Association of Immunologists. Thank you.
Thank you so much, Mary. With this time, we can open it up for questions, and our attendees have been given microphone access. While you're thinking of questions, you feel free to also use the hand raise tool so that we can identify you for questions. Mary, I know you are continuing at RIT for the next year. Is this the research that you'll be continuing, and what are the goals for your next steps here?
Right. Yes. This is actually not the focus of my research in RIT. Here in RIT, I'll be taking it further. I would be looking at the expression profiles of a whole lot of genes, including these, in human samples. I want to look at how can we use these different genes that have different expression profile to, to demonstrate who is likely to have severe condition or who is likely to have a symptomatic condition. And if we see something very interesting, which we are hoping to see, can we use any of these targets as a marker, for diagnosis, for this severe condition? Can we use it as a marker for therapy, for treatment, so that we'll prevent children and other vulnerable individuals from, dying of severe conditions as a result of malaria?
Thank you very much. Do I have any questions? Wait a minute, in case there's any questions for Mary. I do have one more as you were thinking about that. That's the applications of this here are really powerful. So it's really exciting work, Mary. How would you go about sharing that information to be able to put those types of applications in place in terms of identifying the markers? Are there organizations working with the ministries of health or others that you would work with in terms of being able to share this kind of information and developing that type of system?
Thank you, Lindsay. Largely or sentimentally, my studies have really been focused on Africa, particularly Nigeria. And I have a strong collaboration with our National Malaria Control Program and the Federal Ministry of Health, and most of my findings I share with them. But that's really a long way to go because the first step is what we are trying to do is to in silico determine what can we use as a biomarker. Do we have anything that can distinguish individuals that have or that are likely to have severe conditions from those that will not? Once we see that, we'll take it a step further to let's go to the field. Can we get samples from individuals with this condition to look at these several genes, not just TNF alpha? Can we, in other words, we are taking it from the bench top, from the lab to the field. Let us see what is there. So when we see what is there, the next step would be, we have seen this, of course, at each stage we'll publish the findings. Can we partner with biotech companies? These particular markers that we've seen, can we turn it into what, can we embed it in a device, express it with monoclonal antibodies, so that when we do that, we see that and we test it with field samples or field isolates, and it gives us desirable results. We're fine-tuning back and forth and we are satisfied with the result. That's why we can now start talking about policy, talking to the policy makers, talking to different stakeholders that can impact, this is something that we can use to protect our children, to reduce the mortality, in terms of children coming down with severe condition. We can actually use it to detect it faster and then know what to do. Because in severe malaria conditions, you would not only be looking at anti-malaria drug, rather, because the condition has gone beyond the normal malaria affecting the red blood cell, it has started affecting other organs, crossing the blood-brain barrier. You want to look at other associated treatments. So can we detect this on time before it becomes worse? So that's when we start looking at policymakers. But yes, the system is in place, to liaise with those, that would impact and take it to the field.
Great. That's really powerful. I'm really excited to hear continuing how this works for you, Mary. I think you can see we do have a question that was submitted currently. How do malaria drugs work, and will your recent discoveries change the strategy design of malaria drugs?
Yeah, thank you, Adwa. So most malaria therapy, they target the parasites now in the red blood cell. That's why even sometimes the drug itself can somehow cause some deleterious effects. So they target this parasite in the red blood cell. So in a bid to destroy this parasite, they actually destroy the infected red blood cell along with the parasite. So that's why people with severe malaria condition actually becomes anemic. So anemia is a condition whereby they have reduced red blood cells available to transport hemoglobin and oxygen. So that's how the drug works. It targets the parasite. It targets important pathways of the parasite, pathways that the parasite need, for them to survive. For example, one of the drugs, sulfadazine pyrimidamine, it targets what we call the glycolytic, a particular part of the glycolytic pathway of the parasite. Now, this parasite needs that pathway in order to synthesize food and then survive. But the drug targets it. And once that is inhibited, the parasite cannot survive. Now, together with the parasite and the infected red blood cell, they are all taken out by the lymph. They are all flushed out. So that's actually why you have anemic condition in people with severe condition. Now, this study, we set to identify this condition before it does happen, so that in the long run, it will be prevented ab initio. It has, you know, at the moment, we see it as having powerful diagnostic impacts in terms of detecting who can be, you know, who is predisposed to having severe condition. In terms of therapy, in the long run, yes, if we're able to see that, you know, there are multiple, okay, so but before I take this a step further, let me just clarify. Because we'll be looking at multiple genes, multiple transcription factors, it will be somewhat challenging to streamline it to a particular therapy, but it can be streamlined, but it will not capture the whole system. But because we are looking at, we are interested in looking at very important, you know, systems, very important genes that will be expressed. We hope that once we target those important genes therapeutically, you know, we can inhibit the system from progressing further. So yes, in the long run, we hope that therapeutically it can be utilized.
Thank you, Mary. Any other final questions? I think not. So with that, Mary, thank you so much for your wonderful presentation today and sharing your work with us. As you know, we are recording this and we will have it available online afterwards. And so I will share that with all attendees and those who have registered, even if they're not able to be with us here today. So thank you once again, and we look forward to hearing more from you here throughout your time at RIT.
Thank you, Lindsay, and thanks everyone.
And I am going to quickly stop recording.